Cancer. It's a word that strikes fear into the hearts of millions around the world. This complex group of diseases, characterized by uncontrolled cell growth, poses a significant threat to global health. Each year, millions of lives are touched by cancer, impacting individuals, families, and communities worldwide. The fight against cancer is a global challenge, demanding innovative approaches and unwavering dedication from researchers, clinicians, and policymakers alike. Despite significant advancements in cancer treatment, including surgery, chemotherapy, and radiation therapy, challenges persist. These traditional approaches often come with debilitating side effects and, in some cases, limited effectiveness. The quest for a cure remains a top priority for the medical community. Scientists are tirelessly searching for new ways to understand, prevent, and treat this formidable disease. The search for innovative cancer treatments has led researchers down some fascinating paths, exploring the intricacies of cancer cells at the molecular level. One such avenue of exploration focuses on mitochondria, often referred to as the powerhouses of cells. These tiny structures play a crucial role in energy production and cell survival. Emerging research suggests that targeting mitochondria and their intricate relationship with synaptic activity, the communication between nerve cells, could hold the key to unlocking novel cancer therapies. This essay delves into the fascinating world of mitochondria and synaptic activity in cancer, exploring their potential as targets for future treatment strategies. To understand how cancer cells thrive and spread, we need to zoom in on the microscopic world within our cells. Mitochondria, often likened to tiny power plants, are essential for generating the energy that fuels our bodies. They play a critical role in various cellular processes, including growth, division, and a programmed cell death, also known as apoptosis. In normal healthy cells, mitochondria function like well-oiled machines, efficiently converting nutrients into energy through a process called cellular respiration. This process is essential for maintaining the cell's energy balance and overall health. However, in cancer cells, mitochondria often behave differently, exhibiting altered metabolism and function. This deviation from normal mitochondrial activity is not merely a side effect of cancer, it's a fundamental driver of tumor development and progression. Cancer cells, in their relentless quest for growth and survival, hijack mitochondria, exploiting their energy-producing capabilities to fuel their uncontrolled proliferation. In the 1920s, German physiologist Otto Warburg made a groundbreaking discovery that revolutionized our understanding of cancer metabolism. He observed that cancer cells, unlike normal cells, prefer to generate energy through a process called glycolysis, even in the presence of ample oxygen. This phenomenon, known as the Warburg effect, has become a hallmark of cancer. Glycolysis, a less efficient way of producing energy compared to cellular respiration, provides cancer cells with a distinct advantage in the competitive tumor microenvironment. By relying on glycolysis, cancer cells can rapidly proliferate, outcompeting normal cells for nutrients and resources. The Warburg effect has significant implications for cancer development and treatment. It highlights the metabolic reprogramming that occurs in cancer cells, shifting their energy production pathways to favor rapid growth and survival. Understanding the intricacies of the Warburg effect is crucial for developing targeted therapies that disrupt cancer cell metabolism. Section 4. Restoring balance, targeting mitochondria for cancer therapy. The altered metabolic state of mitochondria in cancer cells presents a unique opportunity for therapeutic intervention. Scientists are actively exploring ways to restore normal mitochondrial function in cancer cells, hoping to disrupt their growth and survival. One promising approach involves targeting specific molecules and pathways involved in mitochondrial metabolism. By inhibiting enzymes involved in glycolysis or activating pathways that promote oxidative phosphorylation, researchers aim to shift cancer cell metabolism back towards a more normal state. This metabolic reprogramming has the potential to slow down tumor growth and make cancer cells more susceptible to conventional treatments like chemotherapy and radiation. Another strategy focuses on exploiting the increased dependence of cancer cells on glycolysis. By depriving cancer cells of glucose, their primary energy source, or by inhibiting key enzymes in the glycolytic pathway, researchers aim to starve cancer cells and halt their growth. Section 5. Oxidative stress, a double-edged sword in cancer. Oxidative stress, an imbalance between the production of reactive oxygen species, or ROS, and the cell's ability to detoxify them, plays a complex and multifaceted role in cancer. While low levels of ROS are essential for normal cellular processes, high levels can damage DNA, proteins, and lipids, contributing to cancer development. Paradoxically, oxidative stress can also be exploited as a therapeutic strategy against cancer. Cancer cells, due to their high metabolic rate and altered redox balance, are often more vulnerable to oxidative stress than normal cells. Inducing excessive oxidative stress in cancer cells can trigger cell death, offering a potential avenue for selective cancer cell killing. Researchers are investigating various approaches to harness oxidative stress for cancer therapy. These include using drugs that increase ROS production specifically in cancer cells, as well as developing therapies that interfere with the antioxidant defense mechanisms of cancer cells, making them more susceptible to oxidative damage. Section 6. Synaptic activity, a new frontier in cancer research. In recent years, a fascinating area of cancer research has emerged, focusing on the unexpected role of synaptic activity in tumor development and progression. Synapses, the junctions between nerve cells that facilitate communication in the nervous system, have traditionally been studied in the context of brain function and neurological disorders. However, growing evidence suggests that synaptic activity extends beyond the nervous system, influencing the behavior of cancer cells in various ways. Tumor cells can form synapses with neurons, receiving signals that promote their growth, survival, and even metastasis, the spread of cancer to distant sites in the body. This newfound understanding of the interplay between cancer cells and the nervous system has opened up an entirely new frontier in cancer research. Scientists are now exploring ways to manipulate synaptic activity to disrupt tumor progression and enhance the effectiveness of existing cancer treatments,